Drug pollution poses significant environmental risks, particularly through the persistence of substances like antibiotics in wastewater. The presence of antibiotics in the environment can disrupt microbial communities and contribute to the rise of antibiotic resistance. Drug persists in the environment due to inadequate wastewater treatment. Low levels of antibiotics in the environment drive bacterial resistance. Moreover, antibiotic pollution harms soil microbial diversity and productivity. The complex challenge of drug contaminants in water, particularly with compounds, remains pressing due to the lack of effective strategies or technologies for their controlled removal. Research must be conducted in order to develop comprehensive approaches. This project used amoxicillin as a model for the reaction. It is a widely used antibiotic belonging to the penicillin group. It is normally prescribed for respiratory, ear, urinary tract, and skin infections. However, it is persistent in the environment, contributing to antibiotic pollution and disrupting microbial communities. Lactase enzymes, derived from fungi, are proposed as a sustainable solution for degrading environmental persistence of pollutants. They are natural enzymes found in fungi, plants, and bacteria. These are the key issues aimed to address through this project. Persistent pharmaceuticals like amoxicillin pose significant risks to human health, wildlife, and the food supply. There is an urgent requirement for eco-friendly approaches to manage pharmaceutical waste and mitigate the risks of antibiotic resistance. The objectives of the project are to determine the effects of temperature, pH, and substrate concentration on the degradation of amoxicillin by lactase enzymes. This project used response surface methodology, RSM, to predict and establish optimal conditions for effective amoxicillin degradation. The process began with the preparation of phosphate buffer. The required amount of salts were dissolved in distilled water. pH was adjusted to desired levels of 5, 6, and 7 using HCl or NaOH. Next, the drug solution was prepared by dissolving the amoxicillin in the prepared phosphate buffer. The lactase solution was prepared by dissolving it in the prepared phosphate buffer, and its enzyme activity was checked. In the reaction medium, the drug stock solution was mixed with the phosphate buffer and incubated at temperatures of 30, 40, and 50 degrees Celsius for 72 hours. In this study, 3 independent variables were selected for the experiment, such as pH, temperature, and amoxicillin concentration. Based on the Pareto chart, temperature was identified as the primary factor influencing the degradation process, with pH and amoxicillin concentration having negligible effects. Based on the contour plot of RSM, the interaction between temperature and pH was identified as the most critical combined factor. The study identified optimal conditions for amoxicillin degradation rate of 67%, highlighting the effectiveness of this method in pharmaceutical waste management. What makes this project unique is its focus on biodegradability and transformation, as well as its role in environmental surveillance and as a marker for pollution. The relatively rapid degradation in controlled conditions could make it useful as an indicator for studying antibiotic degradation pathways in natural environments. Amoxicillin's frequent detection in environmental samples, such as wastewater, rivers, and agricultural runoff, makes it a useful marker for assessing pharmaceutical pollution. When it comes to benefits to intended users and society, the lactase-catalyzed degradation of amoxicillin can lead to safer water supplies by effectively removing this common antibiotic from wastewater, reducing the risk of antibiotic resistance development. This process contributes to environmental protection and public health by minimizing pharmaceutical pollution. This project has significant commercialization potential in the pharmaceutical and wastewater treatment industries, offering a sustainable solution for managing antibiotic contamination in wastewater to comply with environmental regulations. Additionally, companies involved in soil or groundwater remediation might explore formulations to target specific contaminants more effectively. Thank you for your attention.